My name's Charlie Nunn. I'm at Duke University in evolutionary anthropology and Duke Global Health. And I'm the director of TRISEM, the Triangle Center for Evolutionary Medicine. I see we have some TRISEM people tuning in. Good to see you guys. This is Club Ev Med. It's Club Ev Med Conversations. We really want to emphasize that part of it, that we want to have a discussion, an active discussion. We choose topics that are really cutting edge in the field of evolutionary medicine, try to highlight those innovations that are happening. In this case, with a paper that has just recently come out, and three authors from that paper. And Club Ev Med is organized by TRISEM, but also with the help of 10 other evolutionary medicine centers and the International Society for Evolution Medicine and Public Health. And I'll paste all that information into the chat in just a few minutes. Before I go on and introduce our speakers, I also wanted to say that we have a series of about 2 or 3 talks that are going to be linked coming up in the next month or so. And this relates to an effort that we're doing at TRISEM involving our Evolutionary Medicine Summer Institute. This is something we hold every year, and we've held about 8 of these now. And this year we have a different plan. This year, instead of it being TRISEM only, we're working with the other centers, the other 10 or so centers that are part of Club Ev Med, to have 2 major changes. First of all, we're going to work with those other centers to organize around a specific topic. And those are the 2 things, to organize it around a topic. This year, the topic we've settled on is evolution-proof strategies for cancer, infectious disease, and vector control. And I'll put that in the chat in a few minutes. And we're hoping to bring in people who are experts and innovators in this area to share with the trainees who come to the Evolutionary Medicine Summer Institute, and also have training modules for those trainees who want to really be leaders in this area of developing evolution-proof strategies. And then finally, of course, we want to build new collaborations across the centers. So this is going to be the first of 3 or so Club Ev Meds that are going to feature some of the cutting-edge research in this area, starting with our talk today. We have 3 speakers today. We have Carlo Meli. He's a professor at ASU. He really doesn't need much of an introduction for this group, especially for those of you working in cancer and evolution. He's at Center for Evolutionary Medicine there, and really a leader in this field. We also have a former PhD student of his, Sarah Sayedi. She got her PhD in 2023, and she's currently a scientist at Anthracis Therapeutics. And then we have Sylvie Hoban, who's an associate professor at ASU, also in Center for Evolutionary Medicine. And she works on host parasite coevolution. A lot of you will know about her work on resistance evolution in particular. And so with that, I'm going to turn things over to Carlo. But before I do that, I wanted to mention that the speakers today are happy to take questions throughout. They'd really like this to be a discussion. So if you have a question at any time, please raise your hand, and we'll try to call on you when we see your hand come up. And of course, there'll be discussion afterwards as well. So raise your hand if you want to ask a question, or put it in the chat. And I'll turn things over to Carlo and his team. Thanks, Carlo.
Great, thank you. It's a real pleasure to be here and to see so many friends I haven't seen for a long time. So thanks you all for coming. We do really want to make this a conversation. So as Charlie said, please do jump in, raise your hand, we'll call on you. And we tried to not give an exhaustive lecture here, but just open up a bunch of topics, because I think this question about how to deal with evolving diseases, evolving systems is a hard one. And we really need the whole community to be discussing and bringing new ideas and figuring out how to translate those ideas to our different fields. So that's the larger project here. I should also mention this paper. I'll have the reference at the end of the talk, but this paper has a large number of authors. And in addition to the 3 of us that are formally talking today, there's a number of the authors are here as participants. I wanted to particularly point out Don Gouge is a pest manager at the University of Arizona and is one of our primary collaborators on this paper, which is really an attempt to take ideas from pest management and translate them to cancer and maybe malaria and other areas as we go. Okay, great. So without further ado, let me see if I can advance my slides. Oh, that's not good. Just a moment. Okay, there we go. All right. So I sometimes give this as a lecture, and this is sort of the way I frame the lecture, that farmers are about 40 years ahead of oncologists.
Oh, no, we can't see the slides yet.
Oh, you can't see the slides. Okay. Sorry.
I can see the slides.
Oh, you can?
I can see
Yeah.
this one. Yeah.
Yeah, I can see.
Okay.
It's just me then. Oh, yep, found them. Sorry.
Okay, good. All right. And this is not meant as a criticism. This is meant as a real opportunity. And what's exciting to me is that there's been so many ideas developed in pest management and agriculture that we may learn from. So I think of this as sort of what are the things we can learn from farmers. So one of the first points is that one of the earliest papers finding pesticide resistance in agriculture was back in 1914. And over the following decades, there was recognition that spraying a field with pesticides selects for resistance. And so this is a common idea that's well known in oncology, that single drug therapies select for resistance. And it's not a surprise to any of the evolutionary biologists or doctors on this call. What's a little bit more striking is that multi-drug therapy also doesn't solve the problem of resistance. And some of you are going to say, sure, but I think if you look at what we do in oncology clinics, we haven't really digested this fact. And I'll try to argue why it's a fact. But here's the references for pest management. So they discovered that combinations of pesticides were not significantly delaying resistance back in the 60s and 70s. And then they started trying to innovate in the 80s, which is basically why I give this 40-year number, that they were facing this problem and this fact that multi-chemical therapies were not working back in the 80s. And they tried drug rotations and some other things. But if you look at the meta-analyses in cancer, comparing single drugs to multiple drugs, the multi-drug therapies do work better than the single drug therapies. So in this case, in lung cancer, across all these studies, on average, adding a second drug increased median survival by about 17%, and that is a matter of months. Same thing. This also meta-analysis looking at 3 drugs versus 2, and at that point there was no benefit found across these studies from comparing 3 drugs to 2 drugs. And essentially you have an issue of sort of diminishing returns from the additional drugs, but increasing toxicity. And so they seem to balance out in the case of lung cancer. In meta-analysis of breast cancer, we found something similar, a 12% mortality reduction from going from single drugs to multiple drugs, and increasing survival by a few months. So it's clear that it's better to use multiple drugs. This is true in gastric cancer as well. Again, there's about a 1-month additional survival from adding multiple drugs. So it's better than single drugs, but it really hasn't solved the problem. We're not getting cures in general from these. So another way of saying that is multi-drug resistance is common. So one of the fundamental differences between pest management and cancer is that the pest managers assume that resistance is present somewhere in the environment. And if you think about that for a moment, that means that cure is impossible. That is, complete eradication is impossible. But if you think about what we do in the cancer clinic, we don't start with the assumption that cure is impossible. And that really, I'm talking here about metastatic, disseminated disease. Because if the cancer is still localized, you can usually cure it from surgery. And we'll come back to that a little bit. So once you accept this or face this, that resistance is present somewhere or very likely to be present, then the goal changes from cure to really control. And I think that's the fundamental shift, and it's at the heart of all of the innovations that came up in pest management. So I think, Sarah, you were wanting to present this part, right?
Yeah, I can go
Yeah.
for it.
So I'll turn it over to Sarah then.
Thank you so much. Thank you, everyone. Yeah, so far as the Carlo mentioned, so the problem it was about the resistance. So and how we can deal with this resistance, because we found that both single drug therapy and multi-drug therapy, we come at the finally we face to the seeing the therapeutically resistance issue. So there is some principle that the pest managers they found it that we call the principle of integrated pest management. So this includes the prevention and suppression. So this is the most important things that we have to first start by that. And then it's going to be monitored. We're going to go over it, so by each of them, and monitor continuously. So that's the other thing, that's defining the intervention and then regularly monitor the tumor burden or the pest, how they respond to the pesticide is the other thing that's really important. And the decision making that includes that we have to be careful about the biology of the patients and the drug and the target of the cancer that's we going to the antigen, or for example that we are going to target. So the choose of the best drug for the patients, for example, or the best pesticides for the pest. And the other thing is about the preferred non-chemical controls over the chemical controls. So that's going to be combination of the other intervention, and that's going to be biological control or the habitat control, mechanical. And the other thing is going to use the pesticides or the chemotherapy or the cancer therapy that's as more specific as possible. And the other thing is going to use the lowest effective dose, because usually we are going over the maximum tolerated dose of that's or we call the MTD set for the patients, but it's better to choose the dose we see the response, but the patient is not going to be show the resistance as soon to the drug. And the other thing is about the choose the pesticides to reduce the cross resistance. So that in this matter, so we are discussing about using the different mechanism of the action. And the other thing is about the evaluate the success based on the long-term management, not based on the short-term response. And the most important things is about their forecast the pest growth and response to intervention to inform most of the other principles that moving forward. So for next slide.
Great. So we're going to take each of these principles one at a time. So this one, starting off really with prevention and suppression. So one of the things that pest managers do is they adjust the crops and also the habitat to suppress the proliferation of pests and also to favor the crop, to sort of shift towards benefiting the fitness of the crop and a detriment to the pest's fitness. So we've recognized in cancer for a long time, actually, that cancer prevention is easier than cancer treatment. Once a cancer has evolved to be invasive and possibly spread through the body, the populations of cells are so large and they're so genetically and epigenetically diverse, it's very hard to control that. So couldn't we prevent it or intercept it before it becomes full cancer? And there is journals in this. There's a division of the National Cancer Institute, although it's still much smaller than the work on treatments. But there's logistical challenges doing cancer prevention. So as you probably know, eating fruits and vegetables and exercising is a good way to prevent cancer, and getting people to do that is very hard. So there's a whole field of behavior modification, how to convince people to live in a more healthy way. There's also issues of time scale, because cancers often develop over periods of decades. So if you want to run a cancer prevention trial on this normal population, you may have to do it over a very long period of time, which is often just impractical. So most cancer prevention studies are done in very high risk cohorts. And furthermore, there's an issue of toxicity, because most precancers or people that are at high risk, the majority of those people, even at high risk, will not go on to get cancer. And so if any of the interventions you do for cancer prevention or suppression have any toxicity, then it can outweigh the benefit of trying to prevent the cancer. One way we're actually looking at this, and some of you may have seen, we just published a paper in Cancer Discovery. We're looking across species and taking a comparative approach to this to try to identify species with very low cancer rates, and then try to understand what are the mechanisms by which evolution has discovered to effectively prevent cancer. So that's just sort of an aside for a fun and really important area of our research. And Zach Compton is the first author in that. He's in the audience right now, and Amy Body, I think, is the last author in that also. She's here as well. Okay, so that's prevention and suppression. The next principle is to monitor continuously and using systems for early warnings. So it's the
Hey,
case.
Carlo.
Yeah.
I wonder if we should ask people if anybody has any comments or questions about the first one.
Yeah,
I mean,
sounds good.
if you don't mind, one that
No, please.
wondering, I was just wondering last week, how much money research funding is spent on cancer prevention as compared to cancer treatment? For example, in the case of smoking, which causes lung cancer, what is the ratio of funding on treating lung cancer versus preventing people from smoking? Do you have any idea what that looks like?
I don't really. It's a little complicated. I know that the epidemiology, which a lot of this cancer prevention sort of falls into epidemiology. I know those studies are extremely expensive. Um, maybe Jay, do you have an answer to this question? Jay
Unmute. You
Leboeuf.
want to unmute.
Sorry, I have another question, so go ahead.
Okay. All right, I'll go get you in a second. So the epidemiology is expensive to have to run these large cohorts to define these effects, and hasn't borne out a lot of progress in many cases. But in terms of like interventions for doing cancer prevention, I think it's much smaller than cancer therapy. But I don't know the
I
numbers.
figured. Yeah, okay.
So go ahead, Jay.
My question is for the last point, I'm interested in studying species with exceptional low cancer rates. I think most people think about that in terms of animals. What about plants? I mean, if you think about plants that are exposed to the sun all the time, we don't see a whole lot of epidermal cancer, I don't think. So are people actually looking broadly beyond animals to see what the genetic mechanisms might be?
Kind of. So Sarah's involved in a project we have looking at what we think is a cancer in cactus. It's called cresting in cactus. In plants in general, there's a phenomenon called fasciations, where the tips of the branches are blow up into these big fans. And we think that's because the meristem cells are getting mutated and they're starting to divide out of control. But there's been very little work on that. It's not even clear yet if there are somatic mutations. So that's the test we're trying to do at the moment on a saguaro cactus. Of course, the biology of plants is very different. And for a long time, people said that plants can't get cancer because they have cell walls that prevent this kind of explosive growth. I don't think that's quite true. And we published a paper in 2015. Athena Actipis is the first author about cancer across the tree of life, where we have some references where people identified 2 cases where there looked like there was metastasis in plants. So that's all to say there's a very little work on that. One of the challenges to getting grants in this is that the reviewers and maybe the funders are not convinced that what we can learn in a plant would translate to better human health.
Thank you.
We're interested.
Great, thanks.
Great. Any other questions? Oh, Michael has something in the chat. Okay, right. Michael's pointing out there's a difference between primary and secondary prevention. So primary prevention is preventing cancer from happening at any time. But once people have a cancer and may have a surgery to remove it or been treated, there's an issue of preventing the cancer from coming back, and that's called secondary prevention. So that's relatively easy to identify that high risk population. And the challenges are quite different because one of the big challenges there is maybe the initial therapy didn't remove all of the cancer, or maybe removed all the cancer but left behind a bunch of pre-cancer that's not morphologically different enough to recognize. And so the approach you take to secondary prevention is going to be different from primary prevention.
And Carla, I want to add to your point, so that and the Michael's points about the secondary prevention. So as also we added in our paper, so about besides the lifestyle, diet, and the exercises, we mentioned also about the controlling the benign tumors, and in the favor of being that to go to the healthy cells instead of going to be more malignant cells, so that we can also say as a secondary prevention, as the Michael's mentioned.
That's right. Yeah, another terminology issue is we have this idea of this term of a benign tumor, which just means a tumor that hasn't yet invaded through the basement membrane. It doesn't mean it's always going to be benign, which is why I have some issues with that term. But there is an interesting question, I think, particularly for us evolutionary medicine people, about how would you change the selective pressures on a benign tumor if you can't just remove it? How do you change them to prevent progression to cancer? And we're very interested in actually just slowing down the rate of evolution as a method of doing cancer prevention. So I'm happy to talk more about that. Great. Right. Bernie is bringing up this point that we can look at what are the mechanisms that are already built into human biology for preventing cancer, one of the big ones being immune surveillance. Right. So as many of you know, immune therapy is what had some major breakthroughs in the last 20 years. And people are asking, can we translate some of that to to cancer prevention to get the immune system to better monitor for these early cancers or precancers and remove them? Great. All right. Let's move on. So the other principle, I think this is actually going to be central to the future of oncology, is monitoring. So there are cases where pest managers or farmers will check the traps in their fields every day to see what kinds of pests are there. But in cancer biology, particularly around therapy, monitoring is expensive. And in the case of something like CT scans, there's a lot of radiation there, so it's actually toxic as well. So that's a practical issue. Another one is that when we monitor with these radiological measures like CT scans, the standard is to measure with what's called RECIST criteria. And the main way we do that is we look at different two-dimensional slices through the body. We find the one that has the tumor, where the tumor is the biggest, and then we draw a single dimension across the longest dimension of that tumor, and that's used as a proxy for the tumor burden. So it's a one-dimensional measure of a three-dimensional complex shape. And it gets worse because inside the tumor, often you can have areas of necrosis, which these measures of just the volume don't take into account. So I'm particularly dismayed with this really poor indication of tumor burden. And that's going to become a problem when later we talk about things like adaptive therapy, which depend on accurate measures of tumor burden to do ideal treatments. So people have been very interested now in cell-free DNA, that's cfDNA, as an alternative. So blood-based tests are attractive because they're less expensive, they're minimally invasive, and you can imagine doing them more often. PSA, prostate-specific antigen, has long been used in prostate cancer and is probably the best blood marker we have in general in cancer. Most other cancers don't have quite as good of a marker you can do, and you can use in the blood to measure the dynamics of the cancer through time. We should also point out that there are big open questions in cancer biology. We don't know how often we need to check the tumor burden, so we don't really understand in general what are the kinetics, how fast does it change. And if you've ever been in a shower that has a long lag time between when you adjust the temperature and when the actual temperature of the water changes, you know that long lag times are a huge problem in controlling a system. So that's going to be an issue for us trying to control this changing system, or changing biology of cancer through time. But I do think that the future cancer therapy is really going to involve better monitoring. And I should point out that the standard of care in most cancer therapies is to give a fixed protocol of dosing of drug, often over a matter of a number of months. And then after some weeks to months at the end of that protocol, to do a radiological scan to see how the tumor is doing. So we're completely blind to how the tumor is responding to our therapies in many cases, which makes it very difficult to adjust what we're doing. Okay, the next principle that Sarah mentioned was this decision making. So in pest management, there's a principle of not actually treating as long as the infestation is below an acceptable level. So they set some amount of crops they're willing to lose, and don't even treat as long as the amount of damage is below that level. And of course, if you don't treat, you don't select for resistance. So in cancer, we don't really know what would be an acceptable level to allow a cancer to be at, and that's sort of anathema to ask, in fact, is because we have such a strong reaction to having a cancer. But there are cases where we do what's called watchful waiting. So prostate cancer is a very slow growing cancer, so we often will wait if it doesn't look very life-threatening. There's precancers like ductal carcinoma in situ, or DCIS, in breast. And there's a movement to, instead of intervening on it, just wait and watch it carefully. So have the patients come back at some intervals to check it. I should say that part of the decision making in pest management is to look at the biology and the ecology of the particular pest. So identify what are its predators, what are its pathogens, what are its preferred habitat, so that you then have levers that you can pull in how you're going to control the infestation. And the sort of one of the analogies here to oncology is that we do take a biopsy of a tumor. Well, of course, the pathologists look at it and tell us what they can from looking at it under the microscope, maybe also with some tests like for estrogen receptor in breast cancer. But also typical these days now is to take a biopsy and send it for sequencing. But what we do with that sequencing is usually just try to see if there's a target, a mutation in the cancer that we have a drug that will address. We don't typically look for presence of resistant cells, of mutations that are associated with resistance, which would negate particular drugs. But I think this is another part of the future of cancer therapy is going to be really delving into the biology of the particular tumors in terms of how they're going to respond to the drugs. Another principle in integrated pest management is to prefer non-chemical control methods, instead of drugs, using mechanical, biological, and habitat controls. And I'll talk about what we mean by that. I should mention here that the pest managers do caution us that you can't get away from chemicals entirely, that chemicals are an important tool in their toolkit. But to avoid resistance from really blooming and making the pest unmanageable, they use these other methods as well. Yes, Cynthia's bringing up something that I wanted to touch on here, which is that different life stages of pests have different vulnerabilities, and that's part of understanding the biology of the pest and what you can do about it. Let's see. And Ford is bringing up the issue that in weed management, they can reproduce so fast, so many seeds, that it's very hard to accept some sort of low level of weeds because they can bloom so quickly. Okay, so for mechanical controls in pest management, that's things like just physically keeping the pests out or physically removing the pests. In cancer, of course, we do a lot of surgery to remove the tumors. And if their tumors are localized, you can get cures that way. There's even some evidence in metastatic cancers that removing both the primary and as many of the mets as you can, that you can do better in some cases. It's been shown in colorectal cancer if there's metastases in the liver, then that you can remove them and actually get better clinical outcomes that way. Pest managers will use pheromone traps, and we could start brainstorming about what a pheromone trap might look like in cancer, but there is chemotaxis, that is cells that are attracted to particular chemical signals. So you can imagine maybe trying to intervene to prevent more metastases by putting in some kind of filters with pheromone traps into the circulatory system. Biological controls are probably easier for us to think about. So in the case of pests, we're talking about the predators, bringing in predators of the pests or pathogens of the pests. One issue that happened early on in pest management, as I understand it, is that the pesticides were also poisoning the predators of the pests. But in the case of cancer, as I mentioned earlier, immunotherapy is now a big player in this field. And you can think of the immune system as predators of the cancer cells. And I should also mention that immunotherapy actually didn't make much progress for many decades when people were just trying to stimulate the immune system. It was only when we addressed the ways in which cancers have evolved to evade the immune system and interfered in those mechanisms of invasion that we made really great progress. So the most effective drugs are these immune checkpoint blockades. So they're drugs that interfere with the signals that cancers are using to shut down the immune system. So then the immune system can then react to the cancer. There's also been some efforts in pathogens of cancer, so viral therapies. So there's some viruses that can only replicate in cells that are deficient for p53, which is the biggest tumor suppressor, or the most important tumor suppressor gene we know of. So it would be a good way of targeting cancer cells and not affecting normal cells if you can give it a virus that will kill cells that are missing p53. There's been a little bit of work in bacteriolytic therapy, that is putting in bacteria that will kill cancers, particularly bacteria that are anaerobic and so can only really proliferate in those necrotic areas of the tumors where it's actually hardest to get the drugs in. So there's interesting ideas of trying to hit the tumor from the outside in the perfused areas of the tumor through drugs that can be delivered to the blood vessels. And then the areas that aren't perfused by blood vessels, perhaps you could inject these anaerobic bacteria that could deal with the cells in that area. Habitat controls, which are sometimes called cultural controls in pest management, are what I alluded to about changing the habitat of both the crops and the pests. So in pest management, that's things like when do you water, when do you plant, and trying to make that different from when the pests emerge. We've talked already about cancer prevention as a version of this. There's also efforts in hyperthermic therapy, changing the temperature of the tissue to disadvantage cancer. There's anti-angiogenic therapy, which means drugs that interfere with the signals the cancers use to grow new blood vessels to feed the cancers. They can sometimes help. They don't work very well on their own, but they can sometimes, in some cases, they're useful in combination with other drugs. And one thing we really haven't looked much at is how do we restrict the limiting resources of the tumor. In fact, in general, we don't know what the limiting resources are for the growth of the tumor. And furthermore, that probably changes through time. So before tumors signal for angiogenic growth of new blood vessels, there's probably a point at which they're hitting limitations of oxygen. After that therapy, maybe it's something different. But we really don't know. And I think there's been interesting work from Troy Day and Andrew Reed that Sylvie's going to mention later, or mention their work later, looking at how by restricting the limiting resources in malaria, you restrict its evolvability and make it easier to treat. So I think that's an area of important potential in cancer that really hasn't been exploited. Okay. Pest managers also try to use pesticides that are as specific as possible to the types of pests. And their main issue there is, or one of the main issues there is trying to minimize the off-target effects on things you don't want to kill. And they also look at drugs that modify pest behavior rather than just killing the pest. And I think that's interesting to ask about how we might translate those ideas to cancer. So one other issue that comes up when you sequence a biopsy from a cancer is they will typically look for the presence of a mutation to target, but they don't necessarily require that mutation have evidence of being in all of the cancer cells, that is clonal, or sometimes we call it in the trunk of the phylogenetic tree or cell lineage of the cancer cells. But of course, if the target is not in all the cancer cells, then you're not going to kill all the cancer cells with your drug, and that drug is doomed to fail. Now, we do have a set of therapies in cancer called targeted therapies that are designed to only work on cells that have a particular mutation that's in the cancer and not in the normal cells. So that would be great because they expect low toxicity. And in general, they have lower toxicity than traditional chemotherapies. Their main flaw is they typically rapidly select for resistance. And I think that's one of the nice things about these ideas from pest management is that the ideas from integrated pest management are all about how to deal with resistance. So this weakness of targeted therapies may actually be okay in this context of using ideas from pest management. Now, I think in general, there's a theoretical point here, which is if you're trying to develop an antibiotic or a pesticide, there's quite different biology between the crops and the pests, or between human cells and bacteria. And so there's lots of targets you can address there that shouldn't have much toxicity for the environment or the host. It's really hard in cancer because the target is a human cell. It's just somewhat mutated from healthy cells. So most of our drugs actually end up targeting all cells that are proliferating, whether they're healthy or not, and which is why most cancer drugs are so toxic. But there is evidence that if you use a drug that's cytostatic, that is preventing cells from replicating rather than killing the cells. So that's like tamoxifen, estrogen therapies in breast cancer, or androgen therapies in prostate cancer. Those drugs it seems to take a very long time often to get resistance to those drugs. It can be 10 years in breast cancer, whereas the cytotoxic drugs that are killing cells, they often select for resistance in a matter of months or a year or two. So I think that an evolutionary approach to cancer should be reviving interest in things like cytostatic drugs and drugs that can affect the behavior of cancer cells to make them less lethal without necessarily killing them and thus not selecting so much for resistance. Another principle here is using the lowest effective dose. And there was an interesting study in lung cancer, and here's the Kaplan-Meier curves from it, where they were actually looking to see if they could get better quality of life from doing palliative care, that is lower dose care, and they compared it to standard high dose care. And not only did they get better quality of life, but they actually extended survival from 9 months to 11 and a half months. And I think this comes from just the basic evolutionary principle that the rate of evolution is related to the strength of selection. So if you reduce selective pressure for resistance, it takes longer to get resistance. And that fits very well with this, what pest managers have learned, which is to use the minimum amount of pesticide as possible to get control. We've done a number of simulations of different kinds of evolutionary therapies in cancer. So this is data from one of our simulation studies, where we noticed that if we capped the maximum dose at below maximum tolerated dose, that's MTD, that there are cases like when we capped it we never went a dose above 25% of MTD, both in adaptive therapy and in standard just constant therapy. This is like metronomic therapy. We were able to keep control of those tumors indefinitely. So the Kaplan-Meier curve here, we had no cases of loss of control. The other thing you can see here is that the adaptive therapy, these dashed lines, were better than the sort of standard therapy in this simulation. There is a principle, of course, of when you do combine drugs, try to minimize cross resistance between the drugs and also separate their application in space and time. So we do this in cancer. We try to combine drugs that have different modes of action. But we actually don't always do that. There are a number of cases where the multiple drugs have been used in you have the same mechanism of action. And even when you do have drugs with different modes of action, you can get multi-drug resistance. And for example, there are efflux pumps that can pump drugs out of a cell regardless of their mode of action. Etienne's asking about cytotoxic therapies. Yeah, so we use this term chemotherapy, by which we usually are referring to cytotoxic therapies that are intervening in cell proliferation and killing the cells that are proliferating. So we get resistance to both kinds of drugs, both cytotoxic and targeted therapies. It's usually faster in targeted therapies, but most cytotoxic drugs, in most cases, particularly in metastatic disease, will generate resistance. Let's see. Good. Okay. There have been ideas proposed both in pest management called negative cross resistance, and Bob Gatenby and his colleagues have proposed a version of this in cancer therapy, I think independently, called double bind therapy, where you might try to combine drugs that select for opposite phenotypes, essentially, such that resistance to one drug makes the cells super sensitive to the other drug. And this has actually been operationalized in pest management. There's cases where the cytochrome P450's detoxification genes in the pest, when they're upregulated, they detoxify one drug, but another drug is provided that's a prodrug. So the cytochrome P450s actually end up activating the prodrug and killing the pest. Separating drugs in space and time is one of the main ideas that actually has been started to be translated to cancer. In many states, there's a mandate that farmers are not allowed to spray all of their fields with a pesticide. And the idea really comes from this observation that resistance has some fitness cost. So in the absence of drug or pesticide, sensitive pests or sensitive cancer cells can outcompete their resistant competitors. So actually you want to keep some sensitive pests around to help compete. Sorry, you want to keep them around to help compete with the resistant pests. So Bob Gatenby learned about this in pest management and tried it in cancer, which in a strategy called adaptive therapy. And the observation in general with our therapies is that we go in with such high doses, we kill all the sensitive cells, and there's nothing left to compete with the resistant cells, and the resistant cells aren't responding to a drug anymore, so we lose control. So Bob and colleagues tried operationalizing this idea of maintaining sensitive cells by trying to keep a tumor stable size over time. So if the tumor was growing, they would raise the dose, or if the tumor was shrinking, they would lower the dose. In the one clinical trial where I'm showing you the data down below, there was a simpler protocol, which is they were measuring PSA levels in prostate cancer, people with hormone resistant, they call it castration resistant, metastatic prostate cancer. If the PSA levels ever fell below 50% of their initial value, they would stop dosing. And if it came back to the original level, then they would start dosing again. And each of these lines here is one patient, and the red is when they were on the drug. It's called abiraterone. The black is when they're off the drug. And what you can see immediately is that some patients had very different kinetics than others. And so this protocol that's conditional on what the tumor is doing actually ends up giving you personalized medicine that adapts to the dynamics of an individual's cancer. And you can see that the adaptive therapy arm, or the adaptive therapy treated patients, had a median time of progression of 33.5 months compared to 14 1/3 months in equivalent patients at the Moffitt Cancer Center. Another point that was really striking to me about pest management was that the pest managers advised to never use the same pesticide twice in a row. And in fact, they try to do it no more than twice in a growing season. And that's extremely different from what we do in oncology. We use the same drug in these protocols every week with occasional breaks for recovery until the tumor grows or until we hit the end of the protocol. So we keep giving the same selective pressure over and over, and we select for resistance. Another important principle that I think is that we have to pay attention to in cancer is to base success on long-term management. So initially, pest managers and farmers evaluated their pesticide based on the crop yield that year and the absence of pests. And often a pesticide will work well for one season or maybe two, but it quickly loses effectiveness. So they learned to shift to looking at the cumulative yield over multiple seasons and the stability of pest control. Now, it's common in oncology to evaluate our therapies based on the tumor shrinkage. We look at partial response and complete response. And if you build a clinical trial around that, you can get your answer much more quickly than if you go for overall survival. But of course, if you go in with very high doses and the highest chance of getting a big response, you also select for resistance the fastest and sacrifice survival. So it's unfortunate. It's logistically a pain in the ass, but I think we have to really base our work on survival time and quality of life. Getting near the end of the principles here, but forecasting pest growth is common in pest management. And if you can forecast it, then you can play out different scenarios of how you might treat the pest or the tumor and develop patient-specific interventions. But to do this, we really need to update our models of what's going on with the data as they're being collected from patients. And the Moffitt Cancer Center has been a real leader in this. They have an evolutionary tumor board where they will work with oncologists and they will continue to update their models of what's going on in the individual patients that are being treated, like weather forecasting. But there has not yet been a large clinical trial of this kind of approach. So this integrated pest management, it really is thought of as a cycle of identifying the pests, looking at how to prevent those pests with those habitat controls, doing the observation of what's happening in the crops and the traps, and updating the prediction models, and then choosing appropriate interventions, whether it's a combination of mechanical, biological, or chemical controls, and then evaluating how those are doing and identifying, are there any new pests, what's going on, and really keeping this cycle going. So it's really monitoring what's happening at a detailed level and adjusting what they do. So this is a summary figure from the paper where we sort of took all of these principles from integrated pest management and gave the examples that we've gone over. So I just put it up here briefly. And then I wanted to mention that there's some other good stuff in the paper that we're not going to go into at the moment. So we discussed what some of the commonalities and differences in the mechanisms of resistance between pests and cancer. So a lot of commonalities, there's a few differences. We also give some examples of what an integrated pest management inspired clinical trial might look like we take colorectal cancer as an example. And so sort of outline how that would go. And then we talk about some of the challenges and clinical needs for being able to translate these ideas to oncology. I've mentioned this issue of being able to monitor. There's also, and not knowing how often we have to monitor, but there's also issues of cumulative toxicity. Some of the drugs we use, toxicity accumulates through time, like with damage to the heart. And if we're doing sort of long-term control, you couldn't use drugs like that. So I'm really excited about this because I think for the first time, we really have a strategy for controlling resistance, which is the major problem in oncology. And this strategy is general to any cancer and essentially any drug, as long as that drug has some effect on that cancer. And so all we really need to do is run clinical trials to compare these ideas from integrative pest management to the standard of care to see if we can do better and change oncology. So it's not the normal 20 to 30 year time scale. It's raising the money and running the 5-year trial. So with that, I was going to want to turn it over to Sylvie to talk about sort of generalizing these ideas to vector-borne diseases.
Well, I don't want to take up too much time because I'd rather have a good discussion, and I can be a little bit brief about this. If you can go to the next slide. So some of you might be familiar with this work, and this is already quite old. This is from 2011, 2013. And this is where I was working with Andrew Reed and Troy Day, and we were trying to apply these same principles, but to malaria and malaria control. And most of it was also around this idea of what is the optimal dose, and lower dosages would reduce the selection for resistance. So if you go to the next slide, super brief, basically what this was a mouse experiment where we seeded a couple of parasites in a sea of susceptible parasites. So just a few resistant parasites against billions of susceptible ones. And then that purple line is showing you what happens if you treat those infections. So they just rapidly get selected, which is what we expect, right? That's why there is an issue of resistance evolution. But if you don't treat it, if you don't do anything, then we barely saw those parasites again, those resistant parasites again. So if you go to the next slide, you'll see what happens if you give less than this aggressive treatment. Then the less drugs you give, the less of an advantage you give to these resistant parasites, right? So it's kind of like demonstrating that effect of the less of a selective pressure you give, the less of a selective advantage you give to the resistant parasites. So when I'm talking with Carlo often about this, how do you manage resistance in cancer? How do you manage resistance in infectious diseases? I always joke that it's way easier in cancer as it is in infectious diseases. I don't stand by that fully, though. But what's happening in infectious diseases and also in agriculture, really, in pest management, is what you're doing today is going to have an impact on tomorrow and 10 years from now and, potentially decades from now, right? Because if you select for resistance, it's going to continue. Whereas compare that to a cancer situation, which is one click. Yeah. So every single individual is going to be a blank slate, right, on an evolutionary scale, because that cancer, by definition, as long as it's not a transmitted cancer, is going to be susceptible to these new drugs. So it means you're losing a drug over the lifetime of a patient, over the treatment lifetime of a patient. But every new patient, you're going to start again, which means that sometimes I wonder with malaria, but in other cases too, like we have this new data, right? Like there might be better ways of managing, but it doesn't really go anywhere. And there's a lot of hesitancy in trying these things for a lot of good reasons too in infectious diseases. But part of the reason is, of course, also is if we've got it wrong, you're going to negatively impact the system. Whereas in cancer, we can try different things, right, within ethical boundaries, and you can see what happens on an individual patient level. And that's on the parasite and infectious diseases. One thing that just completely boggles me is that we have the same questions in vector control, right? It's the same scenarios. There's a lot of insecticide use, a lot of resistance. We know how to do it in pest management, and somehow that never got translated to mosquito control, which is very similar. There's also differences, but it's very similar. So We're still at the stage where we use a single insecticide, usually. There's no rotations, you know, and there's a lot of resistance. So I'll keep it at that, so we'll have some time for.
Great. And I'll just put up, this is the reference to the paper that the talk is based on and some of our grants. Jason was asking about, Jason Samarello was asking about the rates of diversification, both phenotypic diversity and genotypic diversity in pest management versus cancer. I don't think we actually have good data on this. We have some measures of amounts of diversity in cancers, both phenotypic and genetic. Of course, it differs from patient to patient and also from type of cancer to type of cancer. I don't know the data on that in pests. Dawn, do you have anything you can say about the amount of genetic or phenotypic diversity in pests?
It's very different for different kinds of pests. There are some pathogenetic pests that, like aphids, various forms of aphids that just there's no sexual reproduction involved, and so they're genetically identical, their offspring. So, and those are more difficult. They do tend to, as a group of pests, there's lots of species that end up being pestivorous because they're pathogenetic, and resistance challenges can develop very quickly.
So that's interesting. I would have thought that sexually reproducing pests would be harder to manage than asexual.
You would, in some cases that's true, but you end up, I was really addressing it. For some reason, I can tell you that there are many different pathogenic pest issues, but then with insects, sometimes they have conditions where they will actually reproduce sexually or pathogenetically, depending on, yes.
Yeah.
So
interesting. Yeah, Jason, I know that's not a very palatable answer, but I guess the answer is we don't know. There's a lot of interest now in cancers, many of you know, about phenotypic plasticity and how that interacts with the evolution of the cells. And some of that plasticity is controlled by epigenetic marks like methylation that is heritable, and you basically just get straight up natural selection on it. Others not so clear to what extent it interferes with selection versus facilitates selection.
Yeah,
so
thanks. Thanks,
Lucy
Carl.
Woodlawn and I have a talk about that, or a paper about that recently in Nature Reviews Cancer. Sorry, go ahead, Jason.
No, I was just gonna say that was totally palatable, and I think there's maybe more resolution within this where you can start to look at certain types of rates of diversification in pest management as parallel with level of aggression and rate of resistance. So I was just trying to get in my own mind further resolution on that. So that's very helpful. Thank you.
Yeah, we're trying to measure the parameters of the rate of evolution, like mutation rate, population size of the stem cells, generation time of the stem cells, selective coefficients, and heritability, and see if those parameters are predictive of both progression from precancers to cancers or response to therapy and time to resistance. But that really hasn't been done yet. Paul brings up an important case. Paul Frankel brings up an important question about the rate of rotation of different pest drugs and its influence. Dawn can speak to this better than I can. I found like it seems like there was contradictory or at least controversial results of this in pest management.
The rates of change, so it really depends on what insect you're aiming at. If you have a very rapidly reproducing insects in the field, for example, mosquitoes, thinking of Sylvie's work, where you have multiple generations in a season, that's a very different approach to a need for rotation of modes of action compared to a pest where you have maybe 1 or 2 generations in a season.
Is there a general observation that rotations do better than non-rotations?
Oh, definitely, yes.
Yeah.
No, and the more frequently, generally, the better. And there are relatively rare instances where pests actually adapt to rotations. also, but suffice to say, yeah, no, definitely rotations of modes of action, more frequently the better.
Just to add something to that, I think an interesting variant of rotation is instead of rotating in time, you can also rotate in space, like a mosaic treatment where different areas receive different insecticides, for instance. And that works really well too, which I don't know if and how you can apply that to cancer, but that would be an interesting one to think about.
Thanks, Don. Bernie brings up an interesting point. Actually, there's a lot of work on vaccine innovation in the area of flu, and there's a whole council that decides on what's going to go into this year's flu vaccines. Derek Smith at Cambridge has some really amazing results that if you look at sort of evolution and phenotype space of the flu, it's almost one-dimensional, which brings up the possibility of anticipating where the flu is going and putting in to our vaccines, not just what was in the southern hemisphere 6 months ago, but anticipating where it's going. So it's going to depend a lot on the biology, of course, of the thing you're trying to control. But sorry, before we go further, there was a point from Ford, who brought up this paper from Rosenheim in 2018. Do you want to say anything about that paper? I don't remember what's in that.
Or are you still on?
Oh, maybe he's not on. Let's see. Oh, no, he is. Maybe awake from keyboard. We can come back.
Yeah, and we should probably wrap up if you have one or two, last points you want to make.
Ford is saying, let me unmute, so go ahead.
Okay.
Ford, are you able to unmute yourself? All right, I'm sorry.
Okay. All right. I think we should wrap up because we're at the top of the hour and folks are starting to take off.
Yeah.
Thank you, Carla. Thank you, Sarah. Thank you, Sylvie. This was really a fantastic presentation today and lots of great conversation. I just wanted to also reiterate one point I put in the chat. I really hope people will consider coming to the annual meeting of the International Society for Evolution Medicine Public Health. I just pasted that back in the chat. Abstracts are due next Monday. So please consider coming. There's also opportunity to submit symposium applications and things like that. Okay, let's thank
Great.
Carlo and Sarah
Thank you,
and
guys.
Sylvie for
Thank you, everyone.
great presentation. Thanks everybody for tuning in today.
Great to see you all.
Bye.
Thank you. Have a great day.
Bye-bye.